That makes me okay. So welcome everybody to the, I don't even know what this is. It's got to be 7th annual, Angelica, 7th, 8th, 10th? Wow. It can't be right? Maybe, it's close. We're that old. Yeah, maybe 10th annual fossil flow cytometry, fossil flow cytometry, human immune monitoring, couple-day course at Stanford. I'm not sure why everyone's come today. You know, we've maybe come to learn fossil flow cytometry. That's why I put this P-Flow 101 up here, but more and more, and what you'll probably notice from the themes of the talks today, and the experiments you're going to do, at least in Gary's lab, are really turning into, and I should have animated this, but X this out and say SciTOF 101, because I think that's what a lot more people are more and more interested in. And so as you notice, I don't look like a tenured endowed professor. I'm filling in for Gary today because he is off traveling and getting a small award in DC that he wouldn't tell us what the award was, but he said he had to go. He had to show up. But he's probably out evangelizing technology and next generation single cell analysis. That's usually what he's doing. That and, I don't know, talking about life beyond our planet. He likes to talk about that too. I don't know, is Gary going to be back later on this week?
He's going to be here tomorrow.
Oh, so everyone, if you've ever had any questions you want to ask about aliens, been afraid to ask anyone else, Gary's a good one to bring them up to. So I'm going to give you guys an introduction today that really covers, the general ideas behind why I was motivated to do single-cell analysis and single-cell technologies and why I came to Stanford to work in Gary's lab. I've now actually recently left Gary's lab, and I've got my own lab in the Department of Pathology here, doing very similar things, using the same technology platforms to investigate human biology. But I think we're all motivated by the same things, and we've all been able, by the types of technologies that you guys are going to get a bit of an introduction to today. So I'm just going to kind of jump right into it and talk about some of those themes. So when you think about single-cell analysis, I think really, with maybe sequencing being the exception, and only because there's amplification steps that are allowed, really parameters are the currency in that we can't measure many things directly at the single cell level, right? We measure reporters of things. So when you look under a microscope, when you put a sample on a flow cytometer, even in just those two cases, what are we really looking at? We're looking at reporters. So we attach a fluorophore to a probe to measure something that we're actually interested in. And then we measure out the fluorophore, and the level of the fluorophore tells us about the level of the thing that we're interested in inside the cell. And so that's great, and it works really well, and this has been the mainstay of bioanalysis, biotechnology, especially single-cell biology for years. But the problem is accessing the most important cells, the ones we're really interested in, the stem cell populations, the ones that people have spent 10, 20 years navigating a really discrete definition of, it costs us a lot of these parameters. And the problem with that, and the problem that I was having, was by the time you get to the cell you're interested in, so if you're studying stem cells, you get to that cell, I already knew it was a stem cell. Just finding it again isn't really the big part of the experiment. The big part of the experiment is asking a question about what the cell's doing. So, you still need parameters to get at these questions of cellular behavior. And this is particularly problematic when you start thinking about primary human tissues, clinical samples, and a limited amount of material, because you can't do experiments over and over again. So the idea is we need the most parameters we can get so we can not only get to the cell types that we're interested in, but ask questions about what they're doing and how they're responding to our experimental or clinical conditions. So how do we get more parameters? Well, as I alluded to in the first slide, so fluorophores, fluorescence-based analysis. Fluorescence is fantastic. It's extremely sensitive. It's very quantitative. And depending on what the platform is, people have multiplexed this to quite a high level. In some cases of cytometry, up to 17 color flow cytometric experiments have been reported. And I would say when I asked around in Gary's lab when I first came here, everyone here said 12 for the Nolan lab was pretty routine. In my mind, 3 to 5 was routine for me, and beyond that, I don't know. I still have to go ask for help. But apparently 12 is routine. But I'll tell you that kind of one of the secrets up here is even the 17, it's not simultaneous. It's 17 in the same tube, but about half of those are mutually exclusive for one another. And so really even getting into double-digit fluorescence flow cytometry or fluorescence multiplexing analyses, you already need to do experimental backflips to make it work. And all of this, and you'll learn this kind of today in this course, is especially when you start permeabilizing cells and trying to get at things inside, you start doing all these things to biological material, and you get what you might call high background or autofluorescence. And so that can be interfering things too. And the biggest barrier in fluorescence is this, as this spectra points out here, is when you start multiplexing these broad, even in something like a Q dot that's supposed to be a really narrow emission, has still a relatively broad emission, and these emissions start overlapping. And this overlap has to be compensated for. And as you compensate the data, you take away from the quantitative sensitive nature that we love about fluorescence. And so the problem is like to measure more parameters, you need to take away some of the things you like to do. So a group in Toronto came up with Scott Tanner's group. He was in, well, he was at a company, and now then he went to the University of Toronto. And now he's back at a company again. But they came up with an idea. He was an inorganic analytical chemist. I don't know. I think he had a love affair with elemental mass spectrometry. And he came up with the idea that he could take these fluorophore parameters, reporters, and replace them with mass reporters. Really simple idea. But the beauty of mass spectrometry, especially elemental mass spectrometry, is it's got a huge dynamic range. As far as mass spectrometry goes, it's the most sensitive type of mass spectrometry there is. And mass spectrometry in general is really highly resolving. So even telling the difference between something that weighs, say, 170 and 171 is very, very, very routine. I worked on mass specs in grad school that could tell the difference of plus or minus an electron. And so telling plus or minus a proton is like child's play. Take a hydrogen away, add a hydrogen back on. And the beauty of this is because you're not getting overlap, you don't have compensation required. And the other interesting thing is when you start thinking about using not just any element, but non-biological elements, so things you wouldn't normally find in yourself in biological materials as reporters in bioassays. There's almost 100 available, and these would essentially have zero background in your assay. And so some of the things you're going to learn today and learn throughout this course that I'm going to show you is that if you take biological material and put it into these new next generation detection formats, if we don't actually stain the cells for something, the machine looks like it's not working because you can't see anything. So that's good or bad, good and bad. And so how do you get up to almost 100 reporters? So like I said, there's about 100 non-biological reporter channels available. But if you look at the periodic table, there's only about 100 elements total. So where does it all come from? Again, you got to remember I said it's mass spectrometry, right? It's not elemental spectrometry. So you look at something like europium. Europium on the periodic table down here in the lanthanide series has an atomic mass of 151.9, so about 152. But in actual fact, there is no europium 152. Europium 152 is actually about a 50-50 mixture of 151 and 153. And both of those can be simultaneously measured and quantified by elemental mass spectrometry. And if you keep that mindset and go back to the entire periodic table, what you actually have is a whole bunch of uniquely differentiated by mass, stable elemental isotopes that are elements that you would not commonly find in biological samples. And so this is where all the 100 or so reporter channels can potentially come from. So the workflow of our analysis, its single cell analysis, is kind of summarized in this slide here. And I'm not going to go into too much detail of the different components because that's what you're here to learn about at the course. But this is just a general overview. And what happens now in Gary's lab and in a lot of the experiments that we're going to be doing this week, you're going to do things not just about looking at what the cell on the cell surface, but again, we're going to be getting at those questions of behavior. So the first part of the workflow is actually you take your cells and you ask them a question. You stimulate them, treat them with a drug, whatever. In this case, we're going to be stimulating them with cytokines. And then at a given certain point in time, you're going to freeze the biochemistry, freeze the behavior of the cell by chemically cross-linking them. And then the cells, from that point on, it's really like a general fluorescence flow cytometry workflow. So if you've done flow-based or immuno-based staining before, I mean, there's nothing really unusual that you have to do. So we have our cells, we permeabilize them, so we've got the surface and the intracellular epitopes exposed. We come in, with our favorite probes, in this case most of the time monoclonal antibodies, and they're conjugated to these monoisotopic mass reporters. And Matt Spitzer, who's here somewhere, I think, or no, is he here? He's coming. He's on his way. Matt's going to be telling you all about conjugating these antibodies. And before Matt talks to you about that, Wendy is going to be telling you all the nitty-gritty of what you really need to know if you want to start doing large-scale scientific experiments. So I'm supposed to give you the rose-colored glasses, and then you'll have to take them off, and Wendy's going to keep it down to earth. So we've got our stained cells with our monoisotopic reporters, and then the cell suspension is sprayed into the machine, one cell at a time. So through this nebulizer, it's kind of like a flow sorter nozzle, and the cells are lined up, and these little tiny droplets, and you'll get to see this in real life this afternoon, are sprayed into the machine. The cells hit the inductively coupled plasma, one droplet at a time, and what happens is everything's destroyed. So the cells are, the droplets vaporized, the cells are vaporized, all the covalent bonds are broken, and you end up with a cloud of elemental ions corresponding to each cell. And these are temporally separated, but they're very, very, very close together. And what ends up happening is this machine is scanning extremely fast, about 80,000 times per second, and that's fast enough to capture the elemental profile of each cell as it goes into the machine on the order of hundreds of cells per second. And then what happens, just like in fluorescence, the level of each one of the reporters relates directly back to the things we were most interested in the experiment. So the things that their probes were attached to, what they were going after. So in this case, we're looking at a couple T cell populations and stat phosphorylation in response to some question we asked up here. Again, I'm not going to show you guys pictures of what the machine really looks like because you're going to get to see it this afternoon. But this is kind of a schematic of what's going on inside the machine. Is anyone here familiar with mass spectrometry, with time-of-flight mass spectrometry? Okay, doesn't matter. Doesn't matter. This is the easiest form of mass spectrometry to understand. And so the idea of time-of-flight mass spectrometry is that everything you're trying to differentiate things by size. Everything is pushed with the same amount of force in time-of-flight mass spectrometry. And you can imagine, just like if you pushed a golf ball, a baseball, and a medicine ball with the same amount of force, the golf ball is going to take off, and the medicine ball is going to go the slowest. And so the idea is that everything's pushed with the same amount of force up here, and everything flies down this time-of-flight region, and the light things get to the detector faster than the heavier things. Simplest form of mass spec you'll ever have to understand. And the way the machine couples this to the cell-based analysis is that as each of the cells come into the machine, the ions are generated in the plasma, and the ion stream is coming through the machine. There's a set of filters and focusing ion optics that goes through here. But the key is this part. The time-of-flight region is scanning about 80,000 times per second. And that's just like how a laser inside a flow cytometer flashes really quickly. And it flashes faster than the cells are going in. And that way you capture not only the signal of the cells, but the space in between. And that way the individual cell signature can be deconvoluted out of the data stream. And one way, like I said before, we actually tell if a cell's there is we use certain stains that are kind of generic for all cells in the experiment. So in this one case, you'll be doing in one of your experiments, you'll be adding, and we actually add this to every single sample we run on the machine, a DNA intercalator. The DNA intercalator is coated with either iridium or rhodium metal, and those show up nicely in our measurement range. So even if your cell is negative for everything else, you're going to have a positive signal in one of these channels. And what the data actually looks like when you're running, this is what everyone in the lab calls the rain. I used to think it looked like the Matrix, but I kind of got voted out. So we call it we call it the rain. Rachel in the lab actually has a program that can read the files off the machine, and it regenerates a screen, which is kind of funny. So we call the program the Rainmaker. But what you're seeing here, this is actually not the entire mass spectrum, but this is the kind of software summary. And so what the software summary is showing you here, each vertical column is one of the reporter isotopes that we're measuring, and each line, each literally pixel from 0 to 225 here is a single mass spectrum. And this is a visual summary of what the machine's seeing. And so what you can see here, this is 3 cells coming through the machine in the period of 225 scans. And 225 over 80,000 is a very short period of 1 second. So this is still only over a fraction of a second. So what you can see here is on the very left-hand side, this is the 2 isotopes of the DNA intercalator, and you can see negative and positive values coming up for the different markers we're measuring. And the particular markers for the different cells are annotated here. And so a very short period of time, you can see a B cell and a T cell come out, right, where you've got your CD4 T cell markers as well as all your mature B cell markers showing up here. And so this is how the machine actually produces the data, and then the software, kind of real time, goes through and sees these different events or blobs coming out, and each of these are individually integrated and written to a single line of your data file or FCS file that the machine produces. So that's kind of an overview of the tech works. Everybody in Gary's lab knows really well how the machine works. I'll be around somewhat. There'll be a lot of other people around in the lab running your groups. If you've got more questions about anything from the labeling reagents to how the machine operates or how it works, ask everybody. Make them explain it to you because it's good for everyone. They get to learn by explaining and you get to learn more details. But I'm going to kind of talk a little bit more about the experiments and the types of data the machine produces now. So kind of early on, before we went crazy with doing all sorts of different analyses, we wanted to kind of put the machine through its paces. And these are some of the kind of performance features that we figured out before we did the first kind of pie-in-the-sky experiments. So one of the features that I particularly liked about the machine, being a mass spec person, is that it's very linearly quantitative. And so even if you have, say, 4 different channels, or 2 different channels measured at the same time, this is looking at cell standards where I've packed different amounts of metal onto the cells, you get a very linear response across almost 4 orders of magnitude. So what's nice with that is you know that if you're measuring the same reporter and you see twice the number of counts anywhere in the measurement range, you can confidently say that there's twice the amount of material on that cell versus the other cell. One of the other interesting features, if anyone has had to play around with fluorophore balancing before panel balancing, is that generally speaking, for the main 30, 40 mass reporters that we use in our assays, the difference between the brightest and the dimmest metal is only about 2 to 3-fold in order of magnitude. And so you've got your, the brightest ones are right here in the middle between kind of 160 and 170, and the more dimmer metals as you go towards the lower end of the mass range are 139. But this particular experiment is a cell line stained with the same antibody at the same concentration, labeled on all these different mass reporters at the same time. So this was kind of my best approximation of trying to stain everything under the same condition. And so we could have made this signal brighter by adding more antibody, but that would have defeated the purpose of this particular experiment. Now the real data. So what happens when you do kind of a head-to-head experiment? And the way this experiment was designed was when I was starting the lab, again, like I said, I'm like a 3 to 5 fluorescent parameter kind of guy, so I'm not a high-end fluorescence guru. So I went to our high-end fluorescence guru, which was Peter Krutzik, who really kind of standardized all of the PhosphoFlow protocols and made everything work. So it was kind of phenomenal because I walked in and everything in the lab worked already, which is always fantastic as a postdoc. And I said, Peter, what's the limit we can push for a PhosphoFlow experiment? And Peter said, really routinely, he's like 9 colors is what I feel comfortable with. And so Peter designed an experiment that was 7 surface markers and 2 phospho markers and optimized it to run on our LSR2 in the lab. And it took, I don't know, is Angelica here? Anyways, I think it took him and Angelica 3 to 4 weeks, couple tries, fluorophore swapping to get it just right. Peter got it all done, and the data looked great. You can see here on the left, this was the surface staining profile. And then I went to the fridge, and I took out these same 9 antibodies that I had labeled on whatever channels, and I put them in a tube that we stained on the same sample, and I got this data on the CyTOF on the first try. So what we were able to do was, you can't do exactly the same gating scheme, and I did cheat because I actually had 10 parameters because I added CD45 in. Because the CyTOF doesn't have light and lasers, you don't have scatter information. And so sometimes that makes it harder to gate some of your cell populations, especially things like monocytes that tend to kind of pick up a little bit of everything. And so what we did, but this was the gating scheme we came up with. So using that DNA intercalator that I showed you in those previous slides, you can kind of gate down on your single cell populations, drill down, and then here Peter did side scatter by 33. I'm doing 45 by 33. And that gets us our kind of lymphoid myeloid populations. And then from that point on, it's a pretty standard gating scheme to gate out your CD4, CD8 T cell populations along with your NK cells. And so I think if you believe that we did a pretty fair approach with the gating scheme here, then what I can show you is the underlying data gives us almost identical frequencies in both approaches. And not only that, when we drill down on the populations, we got very similar qualities in signal and response. Now, I will point out in these particular experiments, the signaling responses on PE, and I can't remember what the other one was. I think it was 647 coupled phosphoantibodies. They were very good. And the magnitude in this case was actually better in the fluorescence scenario, but the quality of the responses on the same scale was identical between the two experiments. So again, it was very, very, very close. And all of this is in figure 1 in the supplemental figure of the 2011 paper in Science. And so, one kind of higher level experiment, but I thought this was fun too. This was actually the first signaling experiment, big experiment we ever did. This was just an experiment on a Jurkat T cell line. We did T cell receptor cross-linking, and we did a time course from 0 minutes all the way up to about 4 hours. And the point I wanted to make here, and it kind of leads into the next part of my summary, which is some of the applications that have happened, is that even with something that's homogeneous, it isn't always homogeneous. And if you just take this one case in point with a T cell line, and you drill down on one signal here, so the phospho S6 signal around 1 hour, even if you gauge down just on these 1 hour time point, you have very divergent signaling happening. And so this is one of those cases where if you were just doing it, say, by Western blot or any other approach, mass spec proteomics, where you mush everything up in the same tube, you miss all of the divergent biology going on. And so if this is what's happening in a cell line, you can only imagine what's happening in real primary samples. So maybe I've convinced you, maybe I haven't convinced you that it's a good measurement platform. I mean, it's certainly something that's changed the way a lot of people that I've interacted with around here doing their bioanalysis. So what I want to kind of convey to you now is a cautionary tale of data and analysis, in that, when you work really hard to get a new trick working, and all of a sudden, now you've got your data. What are you going to do with it? Right? So if you think about the evolution of flow cytometry, people have, we started back in the early 80s, actually just over in the Herzenberg lab here at Stanford, with a couple fluorophores and some light scatter parameters. So in total, we could make about 6 unique two-dimensional plots. And two-dimensional plots are important because that's how we as people think about data. I can't look at data in a 6-dimensional space. I have to look at it A versus B, and then B versus C, and so on and so on. So as things evolved and more fluorophores and lasers and detectors were added, until we got all the way up to the very first mass cytometry experiment that we did here on healthy human bone marrow, you went from 6 unique plots, now we're up to 500 unique plots. And I've actually done experiments where this is actually up over 1,000 now. And my point being that this is not scalable. So even though every cell you could probably think to look for in human bone marrow is probably in here if you know how to gate it. I'm pretty sure, because we've gone through and manually gated most of this. It requires a lot of prior knowledge, which is scary, because not everybody has 3-4 months to sit down and learn about a new system and learn about manual gating convention schemes to go and get at the cells they're most interested in. And so while it was scary, at the same time, it raised an opportunity, and this has been a huge focus of our group and a lot of other groups, I think, working with these new types of single-cell data sets, not just mass cytometry, is can we come up with cool ways to reinvent the data set or reinvent data visualization? And so the idea is that if each of these are cells or clusters of cells, is that can you take the data and arrange it in a smart way in two-dimensional space that recapitulates some of the high-dimensional features? And I think the short answer, you guys probably already know, is yes. You can make two-dimensional structures that are better reflective of higher dimensional shapes. And so 2 possible solutions I'm going to briefly mention, but they're going to be talked to in more depth by Federico, I think, tomorrow, is Spade and Visney, and both of these have been published on. Oh, and Wonderlust, but this is an old slide. I'm not going to tell you guys about Wonderlust today, but you can come ask me. So Wonderlust is a little more for cell differentiation. What Spade does is Spade puts two-dimensional, our high-dimensional cell clusters into a two-dimensional space on a tree shape, and that gives you some organizational information. And Visney's kind of like a principal component analysis way of coming up with a 2D representation of, again, your high-dimensional shape of your data. And so just to kind of dive into the first example, this is our healthy human bone marrow experiment. You may or may not know it, so this is all published in Science in 2011. And what we did is, I'm not going to bore you with the details of how Spade works, but Spade, again, what it does is it takes your cells, clusters them based on the parameters you tell it. It It does a downsampling so that even rare cell populations like hematopoietic stem cells get a cluster at the same time that more abundant T cells and B cells get clusters. Organizes them with a thing called a minimum spanning tree, and in this case, the size of the node reflects the number of cells in it, and the coloration of the node reflects the expression of the marker of interest. And so in this case, you can go through and do a little tree gazing, and of course, the algorithm doesn't nicely annotate everything. That's a grad student, a postdoc, locked in an apartment for a month, picking out things and making sure you're drawing circles and bubbles right. But the point is the tree really facilitates figuring these things out. And so what you can do is you can overlay the expression of different markers of interest. CD45 somewhat informative because the more mature cells are higher than the primitive cells. But as you drill down, you can pick out your T cells by CD3, CD4 and CD8 T cells. 34 lights up your progenitor cells up there. 19 for the B cell arm, and 20 you get a little more mature cells, and then you come over to the myeloid cells for 33, 11B, and you even have your subsets that are higher, like the plasmacytoid DC cells. This is the high affinity IL-3 receptor, and so on. So it's cool because you've got this model where you've annotated all the cells in the experiment, but we made that model from a pool of samples, right? And from that pool of samples, you can then take one sample, like our basal condition, and subtract the response of another sample, like IL-7. And again, the algorithms can do this kind of stuff automatically for you. So in this case, we wanted to ask the question of what happens to phospho-STAT5 in response to IL-7 normalized to basal. And what happens is when you do a difference tree, all of a sudden the cells that respond to IL-7 on the phospho-STAT5 axis become really obvious. And the same thing for B cell receptor cross-linking and PLC gamma, or even looking at toll-like receptor signaling with LPS and P38 activation. And layered on top of that, you can even add in extra things like drug treatment. So we can look at the effects of something like dasatinib, this is a Src family kinase inhibitor. It's used against pH-positive leukemias sometimes as a second line of defense to drugs like Gleevec. And what you can see here is, well, in some cases, dasatinib, because it targets Src family kinases inhibitors, which are upstream of ERK in B cell receptor cross-linking, it can turn that off in a very predictable fashion. But when you activate calcium ion signaling, ERK in the same cells can be bypassed because, of course, you're bypassing the pathway that this drug is targeting. And the same goes here for something like the STAT5 example that I just showed you. So pervanidate, which kind of generically activates the cells by inhibiting tyrosine phosphatases, what happens is dasatinib can actually, in this case, turn most of the STAT5 off, except in the PDCs. But in something that's a JAK-STAT cytokine phenomena, again, it has no effect. But it's kind of cool how you can not only study normal responses, but taking these types of approaches gives you the flexibility where you can then start adding kind of unknowns on top of that. It's just an idea of how you can design certain experiments. So how has this been applied since? We've got a story that's very close to publication on dissecting oligoclonal AML. So actually taking these cancers that people know by next-gen sequencing are oligoclonal, have multiple mutational species, and trying to overlay functional and phenotypic information on top of that and understand how mutational status might be linked to cell behavior. We've had a nice story, and this is where kind of some of the Visney stuff comes in. And so doing things like coming up with nice 2D representations of data that better reflect the high-dimensional space. And one of the things that Visney is particularly good at is really, what's the best way to say it? Really fatefully segregating unrelated cell types, which doesn't always happen in every dimensionality reduction approach. And so in this case, we were able to routinely separate leukemia cells from healthy marrow, even when the leukemia was very, very rare. Mark Davis's group right next door to us, and you'll be in the Human Immune Monitoring Core on Wednesday hearing from some of them. They've done some phenomenal work on hooking up these metal reagents to tetramers, and so doing T-cell antigen-specific staining. So staining, figuring out like what in this case, they were actually comparing what the CMV cells were doing versus the flu-specific cells, which were over there, versus, I can't remember what the other population was. I think it was rotavirus-specific cells. Not rotavirus, sorry, EBV-specific cells. So it was really cool in that when they drilled down into the same T-cell compartment, I found this surprising because I'm not, as a more of an armchair immunologist, that the same CD8 memory T-cells from the same person, if you could differentiate them based on what virus they had memory against, they had totally different potential to respond and produce cytokines and respond to activation, which I thought was kind of cool. And they've done this kind of one cell at a time, and they've actually also recently upped this to where they're actually multiplexing and barcoding tetramers to the point where they can basically, for a given patient who might have received a rotavirus vaccine, they can screen their T cells for all the possible rotavirus epitopes in one sample. We've also kind of made headway on some of the more equivalency type things. So, some of the deficiencies you've had, things you can't measure with antibodies, like viability, S-phase. We figured out how to do these things with other metal-based reagents, and these were both published in Cytometry. You'll hear a little bit about more of them today. We've figured out how to do longitudinal studies where you're trying to normalize cells not only across long days of analysis, but across kind of multi-days, multi-weeks of analyses, and this is using internal bead standards. And even gone to kind of next generation drug type screens where instead of just doing one drug, like I explained to you with the dasatinib, we've added multiple drugs in, and we've been doing it in a barcoding format. And so what we do is we do these multiplex screens where we can barcode an entire 96-well plate into one tube, and each well of the 96-well plate represents a different drug treatment. And you can come up with these pretty cool summaries if you happen to have an architect in your lab who likes to play around with data visualization. It kind of hasn't stopped there. So there's other potential for developing next generation reagents. One of the routes we've been playing around with for quite a while is of getting more metal on the antibodies is nanoparticles instead of these solution chelators that Matt's going to tell you a little more about this afternoon. And the idea again is this is all playing off of the linear range of the machine. And so the idea is that if you put more metal as a reporter, you can detect less and less on each cell because it requires less copies of your antibody probe to get more signal out of the machine. And last but not least, I've also been playing around a lot with other stem cell systems. So it's not limited to blood and bone marrow. I've been doing stuff with pluripotent stem cells. Another guy in the lab, Eli Zender, has been doing stuff with pluripotent stem cells and reprogramming. We've got projects on ovarian cancer. We've got projects on, Wendy, help me here. What else do we have? I'm trying to think of a kind of non-traditional. Every leukemia you can imagine, I think, has gone onto the machine. But we also have ovarian cancer. We've had some breast cancer stuff. That's about it. Anyways, ask people what they're working on because everyone loves to talk about their project in the lab. And just to kind of give you a glimpse of the future, so like I said, flow's been around for 30-odd years. Elemental mass spectrometry has been around for 30-odd years. So the instrumentation's quite mature. But it's come a long way. So this is a spectra I like to show where this was one of the first inductively coupled plasma ICP-MS papers from 1980. Okay, and using this kind of universal chemistry standard, they were able to get on this one peak 1,000 counts per second. So generally 1,000 ions per second hitting the detector. That same standard run on modern day instruments today gets about 10 to the 9 counts per second. The same universal standard. So in 30-odd years, this technology has improved about 1 million-fold in sensitivity. And so just like every other type of analysis, things are already getting better. But the other thing that I think is kind of provocative is this is a spectrum from the Saitov and Gary's lab of all the reporters that were used for the science paper. And what I want to point out is there's lots of giant open spaces. So on the same machine with the same technology and approaches, there's more reporters. There's more room for multiplexing available. And even this is actually a kind of an old slide, so I apologize, but we've actually already filled in 2 spots here. We filled in about 8 spots right here. We filled in another spot here. And so as people think of new and cool ways to use these metal reporters in their assay, you're able to have more and more multiplexing, and this is where the guilt-free part comes in, without having to feel like you need to take out something else. Because I think normally, in flow cytometry at least, if it's like, oh, I've got to do cell cycle area apoptosis now, well, now what am I, the first question is, well, what am I not going to measure if I'm going to measure cell cycle, right? Where now, we measure cell cycle with a BRDU type analog, except we use IDU, and IDU has iodine in it. And iodine shows up right here, and we don't have any antibodies that we measure on 127, so it's a guilt-free parameter. You just add it into the assay, and it's an extra thing without having to feel bad about losing something in your experiment. The other cool thing that I just want to briefly mention, that's something that recently came out of our lab, is single-cell suspensions are not necessarily required. There's been a couple of papers recently published, one by Baron Boden-Miller's group in Zurich, who Baron was a former postdoc here and has left and is building his own empire at the top of the Swiss Alps, doing imaging with these same reagents. And so Baron's approach uses lasers to sample tissue sections that have been stained with these metal antibodies, and the vapor essentially gets sucked up into a cytoph. We used a similar approach, except we used a different machine that was on campus here, and instead of using the cytoph, we used a machine that uses, instead of a laser, an ion beam, but it's the same idea, scans the tissue section, and the cool thing is you can recapitulate these images. So all of these images that I'm showing you here, these are ductal breast carcinoma tissue sections. Everything here, even though it kind of looks like a confocal or a bright field image, these are all mass spec images. So all we've taken is we've taken the mass spec signal and colored it to look like fluorescence, or colored it to look like your horseradish peroxidase brown precipitate staining that you get on the slide. And we can even measure things like if you get your H&E staining, you can measure like the aluminum that gives the H&E its blue color, and you can recapitulate the blue color on the slide. We can find nuclei using things like a double-stranded DNA antibody, and you can measure more than 10 markers at a time simultaneously. In our paper, we did 16. Baron's paper, they did 30 because they had a cytoph they were reading that on. And so there's lots of things possible. You just think about what happens when you start taking light-based reporters out of your assay and adding in multiplex mass-based reporters. So anyways, yep, lots of cool images. Image segmentation, and guess what? What do we go back to? Parameters A versus B, because what do? We don't know how to see in more than 2 or 3 colors in 2-dimensional space. But my point is there's lots of opportunity for new ways of measuring and multiplexing things, and there's lots of opportunities for bioanalysis on top of that. So I just want to give everyone a broad overview, and there will be lots more details to come. Feel free to ask lots of questions while you're here. Everyone in the lab is very informative and has their own expertise, and if they can't answer your question, I guarantee you they will point you to or pan you off on someone who can definitely answer your question. And we got a few minutes for questions, but if not, thanks for being here so early and listening. Yeah. Bill.
So, what's the smallest particles you've measured? Can you show the vesicles, and what's the largest cells?
Oh, on the cytoph?
Yeah.
The smallest cells I've measured. It's probably the smallest particles. Where's Gojun? Gojun, how small were the silver nanoparticles you put on the machine? So the problem is it's not a particle size, it's a particle signal, right? So Gojun has been doing stuff with next generation reagents. So when he puts a nanoparticle chunk on the machine where every atom in that chunk can be seen by the detector of the machine, it's no problem measuring something that small. Now, I don't know, if you had something else that was that size but it was made out of biological material, I don't know how easy it would be for the machine to see that if you couldn't get the right kind of staining footprint onto that. Does that make sense? Yeah.
So you got that signal. Not on large size. Can you do megakaryocytes or adipocytes or
I would. Well, the upper limit is the nebulizer. So the nebulizer right now, even though they tell us the nebulizer is 100 microns in terms of the diameter, I think the nebulizers, we have ones that are good for bigger cells. I think some of the ovarian cancer cells are almost, they're close to 100 microns themselves, aren't they? Yeah. And we have all kinds, sometimes we clog the nebulizer up, but the take home is as long as you can spray it onto the machine, you're doing all right. You can get nebulizers with openings of at least 250 micron that I've used on the machine before. So I would be more concerned not about the machine handling the size of your particle, but of you, just like in flow, like clogging up your nozzle and your fluidics. But as long as you take the right considerations, I think your size range of particles is pretty wide.
Can you do anything with live cells?
As far as putting them on the machine? Yeah. We have run live cells on the machine before. A couple considerations. So, you can run them on the machine. The problem is, of course, the machine still kills them. You can't get around that part right now. And if you run live cells on the machine, you need to run this machine in an isotonic solution, like VaxBuffer kind of thing. And right now, for maintenance issues, we run the machine basically in DI water. So all of our samples, the last step is we add our intercalator in a phosphate buffer, and then everything's refixed and washed a couple times in water. And we spent a lot of time actually figuring out, because I was convinced that that was going to be really detrimental for the antibody staining, but it turns out it didn't really make any difference if you refix everything and everything's dead anyways. And it keeps the machine a lot happier, because you imagine you're not, because unlike a flow cytometer, there's not a waste bucket full of used sheath fluid, right? Everything you're spraying into the machine goes into the machine. So ideally, you don't want to be spraying cups of salt into it with every experiment. But yes, we have run live cells, and it's totally possible to do. It's just you have to reconfigure the standard operating procedure a little bit. All right.
I don't know if this is this switched on or not.
I think he'll unmute you.
I'm